We are going to finish up this chapter looking at the efficiency of ATP production in both the electron transport chain and cellular respiration, and also in fermentation. So we're going to start with this reaction, which is the classic reaction for cellular respiration, taking in glucose and oxygen from our surroundings and giving off carbon dioxide and water and energy in the process. So how much energy is actually produced is what we want to look at. So the difference in the energy content of the reactants and the products, this exothermic reaction of giving off energy, is 686 kilocalories. In the phosphate bonds of ATP, each one has 7.3 kilocalories of energy held in that bond. If we have 36 ATPs that are produced, and each one has a production of 7.3 kilocalories, that is a total in those 36 ATP of 263 kilocalories total. And we notice up here that the total production of energy is 686 kilocalories. The energy that's held in those 36 phosphate bonds here of ATP only accounts for 263 kilocalories. So we do the math and we notice that there's actually a 39% efficiency of energy capture through this process of cellular respiration aerobically. The rest of the energy is then lost as heat to the surroundings. So let's take a look at fermentation. What happens if we don't have oxygen available and we still need energy production to occur? Well, this can still occur, but not on as efficient of a rate as the electron transport chain and cellular respiration producing ATP will occur. Animals are going to go through fermentation and convert pyruvate to lactate or lactic acid. Some bacteria do this as well, and those bacteria are the ones that we use to make yogurt. Other organisms convert pyruvate to alcohol and carbon dioxide, and these are single-celled organisms. Fermentation regenerates NAD+, and it keeps glycolysis and substrate-level ATP synthesis going, and it has its advantages and disadvantages. It does provide rapid burst of ATP without the presence of oxygen. It provides a low but continuous supply of ATP when oxygen is limited, and glycolysis is the only thing that can function in cellular respiration. Lactate, though, is potentially toxic to human muscles, so it lowers the pH and it causes muscle fatigue. So this chart looks at fermentation. Fermentation, we put in glucose and 2 ATP and 4 ADP and 2 phosphate. The outputs, however, either lactate or lactic acid, alcohol and carbon dioxide, 2 ADPs and 4 ATPs. Again, a net gain of 2 ATPs. Put this all into perspective, cellular respiration is going to start with glycolysis. It's taking the glucose molecules that we eat and in our food source and breaking it down to get energy for us to be able to use, for our bodies to be able to function. So we're going to go through glycolysis and break down that glucose molecule. If we have oxygen available and we're using oxygen, it's aerobic. Then it's going to go ahead and go into the prep reaction, the Krebs cycle, and the electron transport chain with a net gain of 34 to 36 ATP molecules. However, if oxygen is not present, it becomes anaerobic or without oxygen. And this is called fermentation. Fermentation is not as lucrative as what the Krebs cycle and electron transport chain. However, it is a source of ATP, even though there's only 2 ATP being gained in this process. That wraps up this chapter with cellular respiration. I understand that it's a tough chapter because there's a lot of chemistry involved, a lot of biochemistry involved. You'll understand better as you take more advanced classes in college, but this just scratches the surface of the whole idea of cellular respiration so you can understand what's going on inside your bodies every time you take in food and need it for your body to produce energy.